Okay, so today we will cover chapter 17, Genome. Chapter 17 is composed of the 5 subjects, starting from the genome can be sequenced rapidly. Finally, we will cover the proteomics. Okay. First thing is that first subject is genome can be sequenced rapidly. Nowadays, we use high throughput sequencing. So this is from this slide and next slide. I will explain the principle of high throughput sequencing. Okay, this is very simple. Okay, so nowadays the DNA is purified. Okay, so DNA is cut into the small fragment is like this. Okay, so this is a DNA. DNA is composed of the double strand. After this fragment, so we hit the temperature. After that, this double strand is become the single strand. Okay, so fragments are denatured. So we usually use the heat. Okay, after that, some adapter sequence is added at each end of the fragment. So I will show you what is adapt in next slide. Okay, after that, this single DNA fragment are attached to the solid space is like this. So this is the first step in the high throughput sequencing. Next one is that each fragment is amplified by PCR. So this is how to do the PCR procedure. Okay, so usually we added adapter. So this is the universal adapter. We already know this sequence. So it means that the primer is the same. So each fragment has an adapter. The adapter sequence is the same, but it means that the primer is also same. So each where we added primer and DNA polymerase and 4 nucleotides, but these nucleotides tagged with different fluorescent dyes. For example, so guanosine is blue, thymine is pink, on cytosine is is also blue, and this is the adenosine is another color. So we added universal primer and DNA polymerase, and after that 4 nucleotides tagged with different fluorescent dyes. Okay, so after that, so for example, in this case, the first sequence is the cytosine. So in this case, 1 nucleotide, guanosine, is added to new DNA strand. After that, unincorporated nucleotides and unincorporated DNA polymerase is removed. After that, this guanosine, because they have the fluorescent dyes, tagged with the fluorescent dye, so newly added nucleotides is detected by camera. So usually this procedure, this cycle, is repeated 100 or 300 times. So this is the second step of high throughput sequencing, PCR. Okay, the final step is that after that we detect each sequence. For example, this is G, and then in this sequence with T, A, T, T. After that, it will produce a set of partial sequence is like this. They made the set of partial sequences like this. Okay? So at that, we have to assemble this sequence into a series of overlapping fragments, like this. For example, in this case, lead 6 has ATT, CA, TG, AA, GG, GCA, and also lead 5 they have overlapping region. GAA, GCA is like this. And also between the lead 5 and the lead 12, there is also another overlapping region. ACG, GACT is like this. Okay, so assemble the sequence lead into a series of overlapping fragments is like this. And if that overlapping region is removed, at that, we have the single assembled sequence is like this. So this is procedure. You have to know to solve the exam. You have to know to solve the homework. Okay, very important. Okay. Okay, so this is finally we can sequence this full sequence is like this. Okay, so this is usually the people nowadays people used this method to sequence full genome. Okay. Another method is RNA sequencing. So we call it as RNA-seq. Everything is the same except for this step. So RNA is single-stranded, and the RNA is cut to the small-sized fragments. Okay, if that fragments of RNAs are reversed, transcribed to the cDNA, this cDNA are sequenced like high-throughput sequencing. So previously we learned that how to do sequencing. So just after denature, and after add the adapter sequence, after single DNA fragment, cDNA fragment are added into the solid space. After that, the PCR is conducted. After that, assembled sequence is like this. Okay, so RNA-seq is that use the RNA, and everything is the same except for the reverse transcriptions. So fragments of RNAs are reverse transcribed into the cDNA. After that, we did the high-throughput sequencing. After that, sequence is aligned, is like this. Okay. So nowadays people use open the new subject, comparative genomics. Okay, comparative genomics. Because nowadays we have lots of information. So if you are interested in this field, you can major in the bioinformatics. Nowadays, lots of people are interested in this field. Okay, so for example, my friends made some company. So if you see that Standime is the bioinformatics. So the main Standime. Okay. So Standime is a bioinformatics company. Okay, so Their team is composed of a lot of people. So I worked with them in the Samsung. Okay, so they hired a lot of people, AI engineer, and so they used this information. They used this kind of information to make drug, to make a new drug. Okay, so if you see that there are lots of people is in the company. Some of them is the AI engineer, and some of them is the biologist, and some of them is chemist. Okay, so if you are interested in these fields, you can measure the bioinformatics, and you can use this information here. So we have now we have lots of the genomic data. So people use the people make these fields comparative genomics. Okay, for example, compare genome within species or across species. So we have lots of the information. So this information provides information about sequence. For example, we can compare different organisms, human, the rat, or fish, frog, or we can compare the closely related species, human, chimpanzee, so orangutan is like this. And also we can compare within one species, within human, within the human, we can compare each person. Okay. So based on comparative genomic data, we can trace evolutionary relationship. So based on this data, we can make that human is belong to the primate, and this primate is very close to the monkey, and monkey is also close to another rodent, and the rodent is close to another avian. Avian is like this. So if you are interested in these fields, you can use DNA information, so you can make this relationship, or you can get a lot of information is like this. And you can also use this information to develop a lot of new drug. Okay. Okay. Okay, this is one example of comparative genomics. For example, the tiger. Okay, so tiger and lion and some leopard. So this is belong to the cat. They are nearly similar to each other. So this is, we call it as a big cat, tiger, lion, and the snow leopard. So we have the small cat in our home. Okay. So usually we can take the blood sample from this organism. After that, we can isolate the DNA. After that, we can do the high throughput sequencing. So now, in this case, they compare the tiger genome with genome of the domestic cat. Every person, or maybe more, many students have the domestic cat in their home. Okay, so first of all, they compare the genome of the tiger and the genome of the cat, domestic cat. After that, they found that 95.6% is their similarity. So there is similarity between the cat and the tiger, or more than 95%. So based on this value, so they say that they predicted that the last common ancestor of the domestic cat and tiger was the 10.8 million ago. So it means that there was common ancestor. Okay, at that 10.5 million ago, they are divided to 2. So one became the domestic cat. One become the tiger. It's like this. They are separated like this. Okay. So based on this information, we can predict their last common ancestor. And also another case is that they compare the genome of the big cat. For example, they have information of tiger, lion, and the leopards. So they compare the genome, this one with genome of the dog, human, and mouse. So they found that the big cat, the genome has around 1,300 mutation compared to the human or mouse or dog. So they can make this diagram like this. Okay, for example, human and mouse, they are very close. Okay, and tiger and cat they are close like this. Okay, so based on this information, we can make we can trace the evolutionary relationship is like this. Okay, so this is another example of comparative genomics. Okay. Okay. Now is we can go to the second subject. Prokaryotic genomes are very compact. Okay, so before go to the second subject. Okay, so the important thing is that if you are interested in bioinformatics, okay, nowadays you can do major in this field, okay, and your salary will be very higher. Okay, so if you become the AI engineer, you can use this information. You can become the bioinformatics. Your starting salary will be very higher compared to the researcher. Okay. Okay. Okay, the second subject is now we will cover prokaryotic genome. So usually the prokaryotic genome is very compact, very small compared to the eukaryotic genome. So there is another the genomics. We call it as metagenomics. Okay, so for example, the metagenomics people can isolate the DNA from environment in the soil. They can collect the soil that they can do the sequencing. Okay, so in the soil or in the large intestine, we have lots of the microbiome. Okay, Doyon's intestine, there are maybe 10,000, more than 10,000 microbiome exists in your intestine. Or in the soil, there are lots of the microbiome will be located here. So we can isolate the microbial DNA and do sequencing. After that, we can find abundance of the microbial species in the soil or in the large intestine. And also we have lots of information, so we can identify several metabolic pathways present in the sample. It's like this. So this field, we call it as metagenomics. So previously, people used the eye. Okay. For example, there are several microbiomes like this. So people use microscope, and people see their shape. After, based on their shape, people decide that this belong to something like that, something like that. But nowadays, people don't use that method. People use this metagenomics to say that this is E. coli, but this is another, this is like this, okay? Metagenomics. Okay, this is new field, okay? So if you are interested in these fields, you can measure in this metagenomics, okay? And also, to study prokaryotic genome, people used transposon. So what is transposable element? Transposable element, we call it as transposon. Okay? So transposable elements are DNA segments that move from one place to another place. Okay, so this is the Nobel Prize winner, Barbara McClintock. Her name is Barbara McClintock. So she received Nobel Prize in 1983, but she found this phenomenon in the 1940. In 1940, so at that time we don't know the DNA, but she found this phenomenon. One cone, okay, one cone has lots of black, but the other cone, some of them is brown, some of them is black. Okay, so based on this information, he thought that maybe there will be some element we can move is like this. So this is the transposable element. So transposable element can move one place to the other place in the genome. Okay, so this is one example. There are several transposition mechanism by transposon, but I will cover only one mechanism. We call it as replicative transposition. Okay, so they have the transposon element. Okay, so this element remains at the same place, but the important thing is this element is copied and inserted another region. So they are composed of A gene, B gene, C gene, D gene, E gene, F gene. In this case, this component, transposon component, move to this F gene. So it means that this F gene will become the abnormal mRNAs. So they will make the abnormal changed mRNAs. For example, in this case, usually the brown color is like this. However, in this cone, some transposon is moved to here, so they will make the different colors like this. I try to explain very simply. But this is we call it as replicative transposition. Okay. So people use this method to study prokaryotic genome. Okay, so using transposon, okay, so people use the transposon to mutagenesis another organism, so to determine the minimal genome. Okay, so this is the one example, Mycoplasma Genitalium. Okay, so Mycoplasma Genitalium is sexually transmitted pathogen. Okay, so this is a sexually transmitted pathogen. So they are bacteria. Okay, so this Mycoplasma Genitalium has around 483 genes. So in this picture, they only showed 2 genes is like this. 2 genes is shown. A gene and B gene is like this. So usually the transposable element insert randomly into one gene. So in this case, they randomly inserted gene A. So in this case, transposon is randomly inserted in gene B is like this. Okay, so they made the mutant. Mutant is like this. After that, each mutant is put into the growth medium. Okay, growth means that this gene A is not essential. No growth means that this gene B is essential. So based on this experiment, we can determine the minimal genome. So gene B is essential, gene A is not essential. Okay, so people use the transposition method to determine the minimal genome in prokaryote. Okay, do you understand it? So you have to understand this procedure to solve the exam or to solve the homework. Okay? Okay, now we will go to the third subject, eukaryotic genome. So usually our human is belong to the eukaryote. Our eukaryotic genome contains many types of sequence. This is an example of eukaryotic genome. First of all, eukaryotic genome, okay, so we have the chromosome is like this. However, prokaryote, their prokaryote DNA is, they are circular is like this. Their size is very small. So usually our eukaryotic genome average is around 100,000 megabase. And the average of prokaryotic genome is around 10 megabase is like this. It means that eukaryotic genome is very larger than prokaryotic genome. And also, eukaryotic genome have more regulatory sequence. For example, prokaryotic, they also has a promoter, but eukaryote has more, maybe 10 times or 100 times more than the prokaryote. Both eukaryotic and prokaryotic, both of them has a regulatory sequence, but eukaryotic genome has more regulatory sequence than prokaryotic genome. So another difference is that only eukaryotic genome have non-coding sequence, because the only eukaryotic genome has intron, and only eukaryotic genome has the repetitive sequence. So FBI used this repetitive sequence to identify the person. Okay? Okay, so this is another characteristics of eukaryotic genome. They have more. They only have the only eukaryotic has the non-coding sequence. Prokaryotic, they don't have non-coding sequence. Okay. Okay, so this is the table summarize the prokaryote and eukaryote. Okay, for example, bacteria, for example, Mycoplasma Genitalium, previously we mentioned, they only have the 483 genes. And also their genome size is about less than 1 megabase. However, our Homo sapiens is our genome size is 3,100 megabase pair, and we have the 21,000 gene is like this. Okay, there is a big difference between the eukaryote and prokaryote. Okay. Okay, so from this on, I will mention several examples of eukaryote. Okay, first one is yeast. Okay, to make the bread or to make the beer, to make the beer and to make bread, people used yeast. Yeast is the eukaryote. From yeast, they are eukaryote. So yeast is a single-celled eukaryote, but they have the membrane-enclosed organelles, starting from the mitochondria. They have the nucleus, they have the ER, they have a lot of the proteasome, like this. They have the membrane-enclosed organelle. So main difference between the yeast, our E. coli is also single cells, but the main difference between yeast and E. coli, okay, so this is table 17.3, compare the genome of E. coli and yeast. If you see that their genome size is nearly similar, and also their number of protein coding gene is this is 4,000 and around this is 6,000, nearly similar. So proteins with role in metabolism, they are nearly similar. The main difference is the number of genes for protein targeting or protein secretion. For example, E. coli, they are single cells. They don't have the membrane-enclosed organelle. However, yeast, they have even though they are single cell, they have the membrane-enclosed organelle is like this. Okay, so they have to decide it after they synthesize protein. They have to decide it. This protein should be go to the mitochondria, or this protein should be go to the nucleus. This protein should be go to the E. coli, the ER, like this. Suppose they need to define. They have to target. So they have lots of protein targeting, or they have decided where this protein should be secreted. Okay, so they have, for example, yeast, they have higher number of protein targeting or protein secretion. 손세한, do you understand it? 이해했어? Do you understand it? Okay, don't fall asleep, okay? Okay, so another eukaryotic organism is fruit flies. Okay, fruit flies is a very popular model organism for developmental biology. So this is the function of typical eukaryotic genome, especially the fruit flies. So they are composed of the DNA. 26% is they belong to the DNA replication, maintenance, and also 13% belong to the membrane structure and transport, and 13% belong to the cell structure and the organelle is like this. The important thing is that fruit flies distribution of gene function is nearly similar to our human or advanced organism. Okay, so nowadays people use Drosophila to study the arm development or to study our leg development because they are nearly similar to each other. Fruit fly is very small, but their pattern is their developmental pattern is nearly similar to human. So nowadays, people use the Drosophila to study the neuroscience, and also a lot of to study development. People use fruit flies. Okay. Another eukaryotic organism, we call it as Arabidopsis thaliana. Okay, so this is plant. This is the small plant with a small genome. So Arabidopsis, we call it as, in Korean, we call it as 애기장대. So they are pepper, they belong to the pepper. Okay, if you smell the Arabidopsis, the smell looks like the pepper. Okay, but Arabidopsis is that plant. They have a small genome. Their genome size is about 120 megabase. Okay. For example, our maize is rice. Every day we eat rice. Their genome size is the 2,500. Our human genome size is 3,100. But Arabidopsis is only have 120 megabase. Okay, they also have so this is Arabidopsis. So after genomics, we find that around 12,000 genes appears in the they are found in the all flowering plants. They are found in Arabidopsis. They also find another organism, another plant. Okay, but people found that Arabidopsis has the among these 12,000 genes, they have the many genes unique to plants. Okay, for example, they have the genes cell walls and growth. Okay, our human, we don't have the genes for cell walls, and we don't have the genes for water channel. We don't have the gene for photosynthesis, but plant has some specific gene. So this gene is belong to these 12,000 genes. For example, so Arabidopsis has 42 genes. These genes they have function in cell wall and growth. Around 300 genes, their function is water channels. Around the 139 genes is belong to the photosynthesis. Okay. Now we will cover our human genome. Okay, so we cover the plant, we cover the fruit flies, we cover the yeast. Now we will go to the human, our advanced eukaryote. Okay, our advanced eukaryote has a gene family. Gene family means that eukaryotes have a cluster of very closely related genes. For example, in chromosome 16, they have the gene family which is involved in the alpha globin. They have alpha globin gene cluster is like this. And also in the chromosome 11, they have the beta globin gene cluster is like this. Okay. There is a very good example. Subin and Hyuksung, they are very close to each other. They always sit next to each other. Gogayeon and Oyujung is also, they are very close to each other. And they always sit next to each other. And Leia and Marin, they always sit next to each other. They are gene family, they are families like this. Okay? The eukaryote has cluster of very closely related genes. The very good example is alpha globin gene cluster. Another is a beta globin gene cluster. Alpha globin gene cluster, they have delta 2 and this is pi delta 1 and pi alpha 1, alpha 2, alpha 1. And also, for example, if they have the phi is like this, we call it as a pseudo gene. Another promoter. But this pi gene, we call it as a pseudo gene. They lack a promoter. Thus, they fail to be transcribed. So we don't know why pseudo gene exists. We don't know why. But our advanced eukaryote has the pseudo gene. We don't know. If you reveal the reason, you will receive the Nobel Prize, I think. Okay? Okay, so for example, our hemoglobin is composed of two alpha chain and two beta chain. Okay, so it's our adult, we have two alpha and two beta. But when we are in the embryonic status and we are in the fetal status, our hemoglobin, for example, when we are in the embryonic status, so for example, this delta gene and another epsilon gene. Delta gene comes from the alpha globin cluster, and epsilon comes from the beta globin cluster. So they make the delta 2, epsilon 2 is like this. But now, if we become the adult, our hemoglobin only composed of alpha or 2 beta. It's like this. Okay, that's why in alpha globin gene cluster, for example, this gene is actively works during the embryonic status. So at that time, alpha gene is not active during the embryonic status, but after the baby was born, so alpha and beta is actively transcribed. So they make the 2 alpha, 2 beta is like this. So they make the hemoglobin is like this. I try to explain very simply. Okay. Okay. And also we have our advanced eukaryote has a highly repetitive sequence. Also we have the moderately repetitive sequence. Highly repetitive sequence means that repeated thousands of times. Moderate repeated sequence is repeated between the 10 to 1,000 times. For example, STR is belong to the highly repetitive sequence. Short tandem repeats. So what is tandem? In Korean, we call it as 반복. So this is, we call it tandem cycle, tandem bicycle. They are repeated 2 times like this. Okay, so STR, okay, each person has different different number of STR, but basically STR is maybe they are repeated 1,000 times. Okay. Okay, and also, for example, the genes for rRNA or genes for tRNA. For example, this unit is repeated. For example, this is rRNA. So they make the rRNAs like this. But this is repeated 280 times. So our rRNA gene is repeated 280 times like this. In our genome, our rRNA gene is repeated 280 times. This is, we call it as moderately repetitive sequence. Okay. Okay, so this is they compare the type of sequence in eukaryotic genome. For example, promoter is not transcribed and promoter is not translated. So you have to know to solve the exam. Intron is transcribed but not translated. exon is transcribed and translated. So moderately repetitive sequence, they are transcribed, but they did not translate it. rRNA and tRNA is not translated, okay? And also our highly repetitive short sequence, for example, STR, is not transcribed, is not translated, okay? This is main difference. Okay, you have to know, okay? Now we will go to the human biology. So human biology is revealed through genomics. So this is our human genome. Okay, so our human genome, okay, we have the very big size human genome. Okay, the protein coding gene is comprised only 1.2%. The other one, intron, is 26%, and also the other gene is like this. We don't know why. So the characteristics of human genome, only 1.2% is protein coding gene. And also, we only have the 21,000 genes, but we have lots of proteins because another post-transcriptional mechanism, for example, the alternative splicing, we previously learned it, chapter 16. Alternative splicing is from same gene, they can make the different proteins like this. Okay? Even though we only have 21,000 genes, we have more than 50,000 proteins. The reason is because we have several post-transcriptional mechanisms. One example is alternative splicing. Okay? And also, through the comparative genomics, we can reveal the evolution of human genome. For example, after we did the human sequencing, and after we did the chimpanzee genome, after we find the result, people was astonished to find that human and chimpanzee, their genome is 99% is identical. Before sequencing, we don't know. We assume that there will be big difference between the human and chimpanzee. But in genome, there is only 1% is different. How about so long time ago, there was Neanderthals people, long time ago. So at that time, we find their fossil, and we collect their bones, and we did sequencing. Find that 99% is identical. 장성현, because they are 99% identical, we thought that Neanderthals they belong to Homo sapiens, Homo sapiens. Okay, our human is Homo sapiens, and Neanderthal is also belong to Homo sapiens because the 99% genome is the same. Okay, Hyuk-sung and Subin, their genome is 99% is identical. But they are different person, different characteristics. Why? Because every people has a single nucleotide polymorphism. Okay? Single nucleotide polymorphism means that substitution of a single nucleotide at specific position. So one people, they have A in specific position, one people has A, and one people has G, and the other people has T. Okay? Kim Do-yeon and Jang Sung-yeon, they are different. You could see that their phenotype is different. Okay, but 90% of their genome is identical. Okay, but they have their some of their region, the single nucleotide region, they are different. If that, they will make different person. Okay, and also people find that people who has cancer, people who has another disease, okay, and people found that, Lee found that, okay, some cancer patient had some specific SNP. Our normal person, they don't have that kind of SNP. So based on this information, we can say that, for example, after we did the SNP, after we sequenced the Park Soo-bin's genome, we can say that Park Soo-bin has higher possibility to some kind of cancer or some kind of disease like this. Okay? Okay, so if you are interested in bioinformatics, you can major in this field, okay? This information is very important nowadays, okay? And their starting salary is very higher, okay? Okay, so SNP is that single substitution of a single nucleotide at specific position. Each person has different SNP. And also there are several disease-specific SNP. Okay, so by using this information, people make another field, pharmacogenomics. Pharmacogenomics studies how individual genome affects response to drug. Okay, so we are lots of people is here, or about 50 students is in this class. After that, we can collect the genome. We can collect the blood sample, and after that, we can do the SNP array. We can check their SNP. We can check SNP, 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 SNP. Okay. So after that, based on SNP analysis, we can identify those who will respond to a drug or those who will not. So based on the DNA profiling, especially the SNP profiling, people say that this group of people, they will have the good response to one drug. But this group, they don't have some specific SNP. Maybe this group will not respond to the drug. Or maybe this person, if we injected the drug, this person will exhibit bad side effects. Based on the SNP analysis, we can say that. Nowadays, we can say that. Okay, so this is the pharmacogenomics. Okay, study how an individual genome affects response to drug, some specific drug. Okay, so based on the SNP analysis, we can identify the group who responds to drug or who will not respond to drug is like this. Finally, we will cover the fifth subject, proteomics and metabolomics. But today, we will cover only proteomics. So proteomics can provide insight beyond the genome. So what is proteome? Okay. So for example, this DNA profiling, in this case, they check the SNP, single nucleotide polymorphism. But now is the proteome, so this is a genome, we call it as genome. But genome is translated to make the proteins. Proteome is total proteins produced by cell, tissue, or organism at a given time or under defined conditions. Okay? So this is our genome. We have the exon and we have intron. Okay? So this will make the some alternative splicing happens. So they will make the different mRNA. Okay, so different mRNA make the different proteins. Okay, after that, some protein is phosphorylated. So their shape is changes like this. And their molecular mass is also changes. Okay, so this is our figure 17.16 show that proteins of eukaryotic genome proteome. Okay, so our proteins, maybe 20% is involved in metabolism and 10% is involved in transport and more 15% is involved in replication, is like this. And nowadays, people can detect each protein's mass. This is we call it as mass spectrometry. Okay, so nowadays people use proteomics to identify the lots of proteins. So usually I will not explain the in depth, but mass spectrometry can identify the molecular mass of each proteins, and also based on that information, we can identify this protein is what, this protein is what, is like this. So if you are interested in the post-genomics, we call it as proteomics. There are lots of persons is major in proteomics. So if you are interested in these fields, you can apply. Okay. There are lots of possibilities, okay? But whenever I consult with people, about 80% of students say that I want to be a person in the QC and QA. But there are lots of possibilities. You can become the proteomics people, and you can become the bioinformatics. You can major in bioinformatics. There are lots of possibilities, okay? Okay, so do you have any other question today's lecture? So we cover genomes, how to sequence rapidly, and also we cover prokaryotic genome, and we cover eukaryotic genome, and we cover human biology. Finally, we cover proteomics. Do you have any other question? So what was the important thing today's lecture?